Why cloning vectors are used? What is the history of the cloning vector? And what are the features of the cloning vector? And what are the different types of the cloning vector? Like plasmids are there, bacteriophages are there, cosmids are there, bacterial artificial chromosomes, that is also called BACs. Then yeast artificial chromosomes are there. It is written as YAC, not BAC. Then human artificial chromosomes are there. Retroviral vectors are there, which 1976 to 1977 introduction of Maxim Gilbert sequencing method. I have already discussed Maxim Gilbert sequencing method in my DNA sequencing method video. If you have not watched that video, kindly go and watch that video. You will have a very good understanding of Maxim Gilbert method. 1977 report of the first cloning vector, that is PVR 322, that we have already discussed. And tell me about this marker. What are the selectable marker in the comment section? Now, 1978, Nobel Prize awarded for Smith and Albert Nathans for the discovery of the restriction enzymes. This you must remember for the exams. Some exams they ask very means memorable questions, like memory based questions. So you have to ask, you have to remember these kind of things. Now I'll see what are the features of the cloning vectors. So all commonly used cloning vector have some essential features like origin of replication, as we have already discussed. This makes a autonomous replication in the vector. Origin is a specific sequence of the nucleotide from where replication basically starts. So the replication mainly starts from this origin of replication in the plasmid vector. So when foreign DNA is linked to the sequence along with vector replication, foreign desirable DNA also start replicating with the host. So when you will clone your gene of interest, it will also start replicating with this origin of replication. Now what is cloning site? Cloning site is a place where that vector DNA can be digested and desired DNA can be inserted. So this is called the cloning site. Why it is called cloning site? Because it has multiple enzymes and you can choose your interest of enzyme and you just can clone gene of interest inside your multiple cloning site. Suppose you don't have that restriction enzyme inside this multiple cloning site or MCS, you just have to put one restriction enzyme so that you can clone your gene of interest. And how that is done, I will tell you in another video. That is a very interesting topic. Now we'll see about the clone. So basically, what is cloning site? It is a point of entry of analysis for genetic engineering work. Recently, recombinant plasmid contain a multiple uh, cloning site which have up to 20 restriction sites have been developed. Okay. Now you'll see selectable marker. So I have already discussed about selectable marker, I think. So selectable marker is a gene that confers resistance to the particular antibiotics or selective agent. Then why it is selectable marker is used? Because we want to select a particular recombinant plasmid. That's why selectable markers are important. A cloning vector contains a selectable marker which confers on the host cell an ability to survive and proliferate in selectively growing medium. So let me explain it. Suppose you just take a vector and you take your gene of interest. This is your gene of interest, this is vector. And you clone this inside this vector with a particular restriction enzyme. And these two restriction enzymes should be same in your clone vector and your gene of interest. And this is cloned particularly. This is finally cloned inside your vector. Now what is the need of selectable marker? Suppose this is selectable marker. And the next process will be after cloning you have to transform it inside the bacterial cell. Why we transform it inside the bacterial cell? Because we want to increase its copy number. Okay, we want to increase its copy number, and we can isolate and use this plasmid for different purposes. We require a host just like E. coli. So transformation will be done. I will discuss transformation in different video. If you want to learn, please write in the comment section. I will discuss that also. So after transformation, what will happen? We will get the colonies on the plate. Like this is the plate and this is agar plate, and these are different kind of colonies. And suppose this is a this colony is positive, and this colony is positive, and this colony is positive. Then how we will know this colony is positive? Because this plate will contain some antibiotic. Like suppose it is containing ampicillin, and your plasmid will also contain ampicillin. Okay, so only that plasmid will survive which will contain your gene of interest. So that's why it is a recombinant plasmid, we know, because after cloning your gene of interest will be there, and this plasmid will be complete with the ampicillin resistant gene. You will cut it, cut plasmid will not show any antibiotic resistant. When gene of interest will be there, then whole plasmid will be completed and your ampicillin resistance will work, and only these plasmids will be positive. That's why this reporter or gene or selectable markers are very important. Now let us see what are the additional properties of the vector. So it should be short and small, should not be very big. The vector should be very small to insert inside the host cell like E. coli. Compatible with the host should be able to replicate it like plasmids in case of the E. coli. Okay, because plasmids are naturally susceptible and easily uptaken by the E. coli. That's why we use plasmids in case of the E. coli. So vector should be compatible. Okay, should become high in copy number. Okay, so it should be high in copy number. It should be able to express itself utilizing the host machinery. It should be able to move under two systems, that is prokaryotes and eukaryote system. So there should be two mechanisms present, two elements should be present. One can be used in prokaryotes also and eukaryote cell also. That vector will be very useful. Now let us see what are the type of cloning vectors. So there are different type of cloning vectors like plasmid, bacteriophages, cosmids, bacterial artificial chromosomes, yeast artificial chromosomes, human artificial chromosomes, and retroviral vector. So these vectors are used, these vectors are used, and these vectors are used. These are mainly viral vectors. Okay, these vectors are used for the very large chunk of DNA, large chunk of DNA. Okay, and these vectors are routinely used for normal cloning. Okay, now I'll see next. So here you can see this is a multiple cloning site. This is a plasmid. This is selectable marker gene, and this is the origin of replication. Okay, so this is bacterial selectable marker. This is origin of replication. This is multiple cloning site. This is N13 S. This is plasmid vector. This is also having the multiple cloning site, selectable marker, and this is your cosmid vector. This is having the selectable marker, multiple cloning site, origin of replication, and one is cos site. Okay, cos site is there. Then this is your yak vector is there. Okay, and this is tel1, This is tail and this is tail. Sorry, this is tail. Now you'll see this, all this we'll see in detail. Don't worry, we'll see all this in detail. So first one we'll see what is plasmid. Plasmid is basically autonomously replicating circular double-stranded extra chromosomal DNA, which is physically separated from a chromosomal DNA and can replicate independently. Suppose this is a bacteria, it is having this chromosomal DNA, it is having one plasmid. So it is basically independent, autonomously replicating. Okay. So these are commonly found in bacteria. Sometimes they are present in archaea also and eukaryotic organisms also. The size of the plasmids vary from 1 to 200 kb. Most general plasmid may be used to clone DNA insert up to 10 kb. Okay. Many plasmids have high copy number, and high copy number is useful as it produces great yield of recombinant plasmid for subsequent manipulations. The point is correct. Okay. But this point is not totally correct. Why? Because every time when you will use high copy number plasmid, and you will get very high yield of the DNA, and recombinant plasmid will produce high yield of the DNA. That is not the requirement always, because sometimes you need low copy number plasmids also. Why? Because some genes are very unstable. So in that case, you will not need to use very low copy number plasmid instead of high copy number. Okay. So while designing experiment, these things should be kept in mind. Now, however, low copy number plasmid may be preferably used in certain circumstances. I have already discussed. For example, when the protein